Okay, are there any questions?
I have a question real quick.
Okay.
It says there's a worksheet for lecture 2 due on the 4th, which I believe is Thursday, but I don't see that anywhere on Canvas.
Let me look for that. Sorry, you're not seeing my screen here, so give me a minute. I'm looking. I'm not logged into the Canvas website. But now I am. Let me do the screen share.
If you go into grades, it says it at the top.
Just a minute, I'm looking at, it should be in, there's only a worksheet for chapter 3 due this week. You're talking about in grades, there's one for lecture 2. Lecture 2 isn't assigned, it's an optional chapter. And I might not remove the worksheet from that. So you found it under grades, is that right?
Correct.
Yeah, that shouldn't be there because we're not doing chapter 2. Is that answering it close enough? I'm not finding it here, but maybe there's another place to go. Let's go into assignments. No, that's a discussion. So whatever it is for chapter 2, you don't have to do it because that's an optional chapter. I put it up there only because there it is there.
So that's it.
Yeah, let me, sorry, I can't get to it. Oh, I just make it non-readable. There we go. Now you shouldn't see it. You'll have to refresh your grade screen to make sure it's not showing. Yeah, that worksheet is to chapter 2, and chapter 2 is a chapter that I only put up in case you guys are weak on chemistry. And I told you yesterday what terms you should know from chapter 2. I'm not going to quiz you on chapter 2, but I do expect you to know the terms that you should have learned in a previous biology course. Okay? All right, any other questions? If not, we'll continue chapter 1 today for the lecture. Microbial World and You, and includes part of chapter 10. You know, I didn't ask with the bookstore. Has anyone checked with the bookstore? Is the custom edition still on back order? Anyone know?
I looked yesterday and it said it was.
Okay.
Mine is still as well.
Yeah, that's going to be a problem if you don't get it in the first week. I think I've emailed that to the class. So hopefully you guys will get that in the first week. And I'm beginning to worry because I'm going to take the assumption that it's going to take time for once the bookstore gets it before they mail it off to you.
Amazon has it for not the custom edition, but the full amount, even for like way less than the custom amount on the bookstore.
Well, that's good.
Helps.
Yeah, thanks for letting us know.
I got the 12th edition on Amazon for $22 to rent it for the semester.
Oh, that's really good.
I bought the 12th edition for $65.
Okay. All right. Yeah, my guess is that other students in the country use that edition. And actually, if you could figure out the reading assignments, although I've got it pretty much just by chapters, the only one that's by page numbers is chapter 10 because it's only a section of that. So you could get the older editions as well. You just have to figure out what to read. But like I said, I've got it assigned by chapters, so that shouldn't be a problem. The only one would be this one. You'd have to check and see classification microorganisms, which page it's on. All right, so this is what we're going to do today. There is no lab, so we'll only be talking lecture, and that is when we come back for the second meeting, which is what, 2:30. We will continue with the lecture. Any questions about that? This is going to be a very busy semester for you because we're truncating it into 8 weeks. And for those of you who take more than one class like this, it's going to be really difficult because you're getting so much information in the semester over 8 weeks. And we're meeting 4 times a week. Anyone else have a class where you're meeting 4 days a week?
I've got human anatomy, but it's kind of self taught with the videos. It's not live Zoom like this.
Oh, okay, I see. All right, well, still, that's going to be an awful lot in 8 weeks. And you got this in microbiology, so you're not going to have much free time. I hate to say that, but truncating 15 weeks into 8 is really, it's going to be quite a push. So you guys need to apply yourselves, and what you should do is every day, besides the class, review your notes, because otherwise it's just going to be too much for you. All right. Don't forget that the Play-G quiz is due this Friday. And I have all the quizzes. I don't know. I just decided we weren't meeting on Friday, so I put the quizzes on Friday. With the exception of the practice quiz, if you want to take the practice quiz and you get your answers correct, it is a good idea because it'll give you practice for the quizzes. It's quiz 00, and I'll give you 1 point if you get all the questions right. If you get all of them wrong, I'll give you, a little bit of something. But it's not much, but it's a little bit. All right, any other questions? I am recording this, aren't I? You guys should see it.
Yes, you are.
Okay, a little button above your thing. All right, only 16 students today. So don't feel shy about asking questions. There's only 16 of you. All right, let me get out of here. And I'll go ahead and do that. Don't forget about the extra credit assignments. The extra credit study sheet is due June 6th. If you're taking the Play-G quiz, I already told you, the extra credit picture is also due June 6th. Those are 2 extra credit assignments that are very early in the term. I think I sent out an announcement that we were, I was having trouble putting the recorded videos on my YouTube site. So I created another YouTube site, and this one will only be you guys. What I was trying to do is divide the YouTube site up, and I just couldn't figure out how to do it. So this link here will get you to our YouTube site, and the link is kind of funny. It won't open in this page, but you can click on this link above here, and then it opens in another site which goes to YouTube, and this is my site, and there's our first lecture meeting and our first lab meeting. Okay. Any questions about that? All right. Let's go back to the lesson. There's someplace around here. All right, we finished with that slide. I know that's hard to see. If something's hard to see, just let me know and I'll blow it up. Let me see what I can do here.
It's a lot better today. Yesterday, it was just super blurry.
Really? That's odd. I don't know why that was, but there must have been a poor connection. I do have a different connection today, which you probably can tell from the background. Boy, you can see my stuff over there. What is that? Oh, I know. That's my trying to hide my toothpaste, and there we go. We'll go like that. Of course, that shows more my bed. All right. Microbes are numerically the most abundant life forms on the planet. How many microorganisms are there? We're only talking about prokaryotes. We're not talking about viruses because, whoosh, that would be tenfold the number that we'll say. And then prions would add a little bit to that. But you know, nobody knows exactly how many prions there are on the planet. So it's hard to answer that one. 1 gram of soil has about 10 to the 8th prokaryotes. 1 gram, depending on how moist the soil is and how much organic material is in it. Does 1 milliliter, that would be equivalent to 1 gram of ocean water, is that more or less? Anyone want to take a guess? You guys aren't very adventuresome. Okay, how about 1 gram of human feces?
That sounds good.
That sounds good, meaning what? More or less than 10 to the 8th microorganisms in it? Don't want to guess. How about on 1 human being?
It's more, right?
It's more than 1 gram, so yes, more on a human being, and obviously more on the entire Earth. In a milliliter of ocean water, you find around 10 to the 6 microorganisms. A human feces, you find between 10 to the 6th and 10 to the 11th grams, depending on how fresh it is. Can you guys see that now? I can blow that up even further, although it's a little bit. There we go. So 10 to the 11th would be when it just comes right out. 10 to the 6th is after it's been on the ground and dried up. Not totally dry, but dried up. 10 to the 8th, like I said, number of cells in a prokaryote. You have 3.8 times 10 to the 13th bacteria and maybe archaea on or in a human. Okay, so that's 38 times 12 zeros past it. On the entire Earth, now I've got it so small, can't see that. The human population is a little over 7 billion, and I don't remember, is it going towards 8 billion? Does anyone remember? I don't remember where it is. And there are nonillion, meaning 1 times 10 to the 30th prokaryotic cells on the planet Earth. Many, many more times than the population of humans. Okay, let's blow that up. All right. Now I can't get the slide. I got that blocking. Here, maybe I can put it like there. Now that'll block your view. Trying to do these. When we're talking about prokaryotes, you should realize that some of them can cause disease. If a microorganism can cause disease, we have a term for it, and that is a pathogenic microbe. Pathogenic means disease causing. Now, when we talk about pathogenic microorganisms, you should realize that most microorganisms do not cause disease. Only a small percentage are pathogenic. Only a small percentage can cause disease. Any questions about that? The vast majority do not cause disease. Now, when we're talking about disease, it's important to realize that there are two types, and so we have to define them. There are infectious diseases and inherited diseases. I mean, you could define diseases in many different ways and have different terms, but we're talking about infectious diseases because those are the ones caused by a microorganism, and they are the ones that can spread from person to person. And that's why we call them infectious. What are some examples of an infectious disease? I'm going to be quiet here until somebody answers to encourage you guys to participate.
C. difficile.
Yeah, when a patient has a C. difficile infection, that would be a disease caused by a microorganism. Now that's a specific disease that is caused by a microorganism. Let's try and get a disease which doesn't have the same name as the microorganism. Anyone? Come on, somebody knows a disease.
Strep throat.
Yeah, strep throat, caused by Streptococcus pylori. We'll study that in one of our labs. So those are infectious diseases. They're caused by microorganisms. You could say another one which you're reading about in chapter 1, the first disease caused by a microorganism that was studied. Geez, I'm drawing a blank here. What is that called? Started by Bacillus anthracis, and it causes anthrax. You'll read about that in chapter 1. Dr. Koch discovered that bacteria causes the first infectious disease, and it was a good example because it was a very large bacteria and it had structure to it, so it gave it something for him to draw and identify, and he could distinguish it from other bacteria because it causes, well, later on, when growing, the bacteria can form an endospore inside the bacteria. Anyways, you can read about that. The other type of diseases are inherited diseases, and those are diseases caused by a person's genome. They are born with those diseases, and an example of that would be something like sickle cell anemia, Tay-Sachs disease, Huntington's disease, diseases like that. Now, there are other types of diseases which don't quite fall into either infectious or inherited, and we won't really talk much about them. We'll be concentrating on infectious diseases because those are the ones caused by a pathogenic microbe. But the other type of diseases would be those caused by environmental stress or aging, and that would be something like tennis elbow. Tennis players sometimes get it, and that is the stress from playing tennis puts stress on their elbow, and the more they play tennis, the more likely they're going to get tennis elbow. And that's simply because their elbow is exposed to stress of playing tennis. And if they never played the tennis, they probably wouldn't get the tennis elbow. Any questions about any of that?
Is psoriasis kind of inherited or more just something you gain? Because I found that people who do not have it, as in the family, I've had a friend where she just gained it over time. Is that something more skin condition or?
There are diseases which, well, actually, I don't think they know what causes psoriasis, but there are diseases which are both caused by environmental exposure, and it might have been just the stress of, the sun exposure. I don't know about psoriasis. And there's other diseases which are inherited. And then there's some diseases, like, let me think if I can remember one, which there's a mixture where the disease is not totally caused by environmental stress, and it's not totally caused by the genetic makeup. It's caused by a mixture. Like diabetes usually is a mixture, and there's both genetics that play a part in the disease, and there's environmental stress or factors that play a part in the disease. So there isn't one thing that causes that disease. For example, a person who never ate sugar and had a well-balanced meal all the time would be very unlikely to develop diabetes, whereas an identical twin who has the same genetic makeup, if they are constantly eating sugar, then they would be more likely to develop diabetes. All right, is that clear?
Yes, thank
Yeah,
you.
all right. So here we're looking at some microorganisms. Let me shrink that just a little bit to show you the slide. I guess that's only saying microorganisms on it. Showing you that they're small. They come in various shapes and various sizes. How small is the average prokaryotic cell? How small do you think? Actually, it should be pretty easy. It should be showing on your screen. Come on, you guys.
Micro small.
Yeah,
1
no,
micron.
see right there, around 1 micron. And they can be longer than that. Like E. coli is longer than 1 micron, but like when you go across, it's generally only about 1 micron. Most prokaryotes are between 1 and 2 microns. I should say most bacteria, which will be the same for the archaeobacteria. So that's the size that we need a light microscope to see. Eukaryotes are bigger, and their size generally is on the size, and each cell in the eukaryote can be different, but it's about 10 times larger than a prokaryotic cell. So a eukaryotic cell, which is right here, will be about 10 times the prokaryote, so at least 10 microns. Okay, any question about that? We mentioned that, oops, went too far. Microorganisms play very beneficial roles on the planet, like they decompose organic waste. We talked about that yesterday. I didn't talk about there's microorganisms which are producers in the ecosystem. These are bacteria that can engage in photosynthesis or chemosynthesis, and they can make sugar by fixing carbon dioxide and water in their environment to make the sugar. And that's very similar to green plants. So they're very important for the planet, because without the producers, we would really starve to death. I mean, you could eat the cow, but then what's the cow going to eat? And so the cow's going to starve to death, and then we won't have any cows to eat. So you do need producers on the planet. You need a lot of them. And microorganisms are an abundant source of the producers. Of course, green plants are the other abundant source. Microorganisms are involved in the nitrogen cycle. And as we discussed yesterday, almost every step, there were a few that microorganisms weren't responsible for the steps, but most of the steps in the nitrogen cycle happen because of microorganisms performing that step. We use microorganisms to produce many substances for us, such as industrial chemicals, such as ethanol and acetone, which are used in industry. We use microorganisms to make insecticides. And to tell you the truth, I don't know much about that, but I know of one, and we'll talk about that a little bit later. But apparently some insecticides are made by microorganisms, and that's probably better than an insecticide, which is a chemical that humans make. We use microorganisms to ferment our food. If anyone likes vinegar, which most of you probably don't really like it, but you use vinegar, and cheese, bread, beer, that's just a few of them. There's a whole slew of foods which are fermented, like chocolate is fermented, coffee, I don't remember all the other things. There's a lot of foods which are fermented, and those are fermented only because the microorganisms are fermenting them. There are products used in manufacturing like cellulase, and those come about because microorganisms make them. And I'm sure you guys all know that an important thing that microorganisms make are antibiotics, and that's how we can get better after we have a bacterial infection. There are other products used in medicine like insulin, like human insulin now is almost entirely made by microorganisms. And what they did was they took the human insulin gene and inserted it into a bacteria, and then they used the bacteria to make the insulin. The point is, microorganisms are used a lot by humans, and we really need them. Any questions about that? Sorry, I just lost my plate there. I gotta shrink that a little bit. Biotechnology and genetic engineering is the next topic we're going to talk about. Biotechnology is the use of microbes and organisms to produce foods and products for human use, and that is centuries old. Like fermentation of wine happened a long, long time ago. I think when the civilizations were just recently started. Somebody left the grape juice house and bacteria got in it, or maybe it was already in the wine, grape juice, and it fermented, and somebody drank it afterwards, and they said, boy, this is really good. And anyways, I'm not sure what the first use of biotechnology was, but the fermentation of food is really, really old. Not just a couple of centuries, but many centuries old. Now there is a new aspect of biotechnology, and it first began, I think it was the late 60s, but maybe it was the early 70s, where it's a new technique of biotechnology, and it's called genetic engineering, where you insert genes into organisms. And those can be used for many applications, such as producing a variety of proteins from bacteria and fungi. And we use that to make vaccines or enzymes that we need. There's also the application of gene therapy, where you actually put the correct gene into a patient that is born with a defective gene. And that's best been studied with the individuals who have an immune deficiency, and you get somebody else's gene and put it into the patient, and that's gene therapy. And then I think you're all familiar with genetically modified bacteria or genetically modified crops. You can make genetically modified bacteria to help protect the crops from insects or from freezing. And there's other things you can do. Like a long time ago, I don't see it in the stores anymore. Somebody made the Flavor Savor tomato, and it was genetically modified tomato, and it didn't ripen the way a normal tomato did. And so you could ripen it and then sell it in the store, and it didn't age normally. And so it would keep better for the stores. And the stores really liked them, but I think people didn't want a genetically modified tomato, so they didn't buy it. And Anyways, I haven't seen it in the store recently. Their microorganisms are also important in microbial ecology. That would be the ecology happening because of microbes. And we have mentioned that bacteria are responsible for recycling the elements in the nitrogen cycle, meaning they move one aspect of nitrogen and convert it into another form. And that's why nitrogen as well as carbon, sulfur, phosphorus can all be recycled on the planet Earth. It's because of microorganisms doing that. And that's true when an organism dies. The microorganisms decompose it and then make the nutrients in the formerly living thing available for plants and animals by recycling it. Any question about any of that? So those cycles, like the carbon cycle, the nitrogen cycle, the sulfur cycle, those are microbially mediated biochemical cycles that support life on Earth. And if we didn't have the microbes doing that, then we would start running out of nitrogen on the planet Earth because it wouldn't be continuously recycling. And eventually it would be limited, and then life would have a hard time living on the planet. The microbial mediated biochemical cycles were first studied in the late 1800s. And when I give you dates like this, I'm never going to ask you, well, I shouldn't say never. I remember one exception. Generally, I don't ask you to remember dates. So when I show you dates like this, you don't have to memorize it, okay? You should know that Winogradsky and his co-worker, you don't need to know the second name, Bajering, were the first to study the microbial mediated biochemical cycles going on the planet. And I think they first discovered in the nitrogen cycle, they learned that when they were studying Winogradsky columns, which is something named after the doctor who was studying it, that the reason why nitrogen was cycling was because microorganisms were taking one form of nitrogen and converting it to another. All right, any question about any of that? Microbes are important in the planet and for human welfare because microorganisms are important in bioremediation. Bioremediation literally means life and to remedy. So to remedy with life. And that's the use of living organisms to deal with an environmental problem. Like right here, there's an oil spill and they're spraying microorganisms on it to help break down the oil to convert it into CO2 and water, and then that would get rid of this pollutant. And bacteria can be used to get rid of things like pollutants. And we mentioned that it's because of bacteria that we get rid of our human waste, which we call sewage, but we can use them also to get rid of pollutants like an oil spill. And then even things like mercury, a spill like that, we can use bacteria to not really break it down, but convert it into a substance which is far less toxic to us than the current aspect of the current spill of mercury. You can use it to detoxify pollutants and break it down into a less harmful substance. All right. Microorganisms are important in the human welfare because we can use them to make biological insecticides, and they're just less toxic than man-made chemicals. But they're also useful because it's another way to make insecticides. And they can be an alternative to a chemical pesticide, and they're used widely on the planet to help prevent damage from insects to crops and also to prevent disease transmission. One example that I know about, and this one is really old, my father used it in the 1960s. Bacillus thuringiensis is a bacteria that produces, let me see if I can shrink that down and you can see it maybe better. No, it produces something which is an infection which is fatal to many insects, and here it is here. And it's harmless to humans and other animals and plants. So my father took the Bacillus thuringiensis and then he put it on his fruit trees. And immediately after, and for about 2 weeks after adding this, they didn't have any insect damage. Now, that worked perfectly on the cherries, but for the apples, he didn't realize that you have to do it every 2 weeks, and he kind of got tired of doing it. And so he didn't do it every 2 weeks. And with apples, for example, you have to do it the entire time the green apple is growing. And if you miss for any 2 weeks, then the fly can lay an egg in it and you can get worms. And so we got worms because he didn't do it for every 2 weeks, but the worms were much less than the time that he didn't do it. So it did work. It's just that he needed to repeat it every 2 weeks, and he just wasn't committed enough to do that. Anyways, that was available way back in the early 1960s. I don't remember the year because I was very, very young back then, but that was available way back as far as 1960. All right, any question about the importance of microorganisms to the planet or to humans? If there's no question, we're going to move on. We're going to talk about the naming and classifying of microorganisms. And this comes from both Chapter 1 and Chapter 10. And I did point out that you do need to read a few of the pages in Chapter 10. When we're talking about scientific names, you should realize that they were established in 1735. That's the 18th century. You don't need to know that date, but I would like you to know who established scientific names. Does anybody know? You should have had that in a previous class. Nobody knows who this is, or nobody wants to volunteer. To give you a reason for taking a guess, I'll give you an extra credit point if it's right.
Was it Carolus Linnaeus?
Yes, Carolus Linnaeus. Oh gosh, it's down there, you guys. Should have said that. And you can pronounce that in different ways because he had a Scandinavian name. I think it was Swedish. And he liked Latin so much that he Latinized his name and changed it to Carolus Linnaeus. I think it was actually Carl Linnaei was his Scandinavian name. So whoever answered that, can you email me that you answered Carolus Linnaeus and you get the extra credit point because I don't even know who answered. I can't see you there. I've only got 2 students here. And you guys can probably see that. And one person, it's blank. And another person, it's a picture they've added. And let's see, there's one person who I can see. I should put it there because sometimes seeing you guys really helps me know if you're getting it or if you're lost and all that. So I should try and put a picture there. Christopher, do you mind my showing your picture to the class? Because if you do mind, I'll move on to somebody else. You have to nod your head or shake your head. No, you don't mind. Okay. All right. Yes, so Carolus Linnaeus is the first person who came up with the scientific names, and he made it so that each species has two names. And apparently it was such a good system he did, because he went around naming everything, that scientists worldwide adopted his system. And every organism, every species has two names for their scientific name. The first name is their genus name. Like Homo sapiens is the species name, the first name is the genus. And then the second name is the specific epithet, meaning the species epithet. You can't say it's the species name because the species name is Homo sapiens. And that is called the scientific binomial nomenclature, or the scientific names, or the binomial nomenclature. And it's called binomial because every species has two names. And it's used by scientists, so the scientific names. And it's used by scientists wherever they are in the world, whatever language they speak. So Homo sapiens is the name for the human species. And if you use that in any country in the world, it will be understood by anyone who understands scientific names. Any questions about that? All right. So when you're using the binomial scientific nomenclature system, you should realize that all scientific names are either italicized or underlined. Like if I were to write that on the board, I can't write it in italicizing it. I just can't write that way. But I could write it and then underline it. And the genus name is capitalized, and the specific epithet name is all in lowercase. The name is Latinized, so it's always based on Latin and used worldwide. The names generally are descriptive, meaning it says something about the organism, or they honor a scientist like Escherichia coli. That's honoring a scientist. He was an early microbiologist, and it's also descriptive. Coli tells you where it's found. It's in the colon. Homo sapiens is descriptive. Homo means, I believe, man, and man that walks on two legs, and sapiens, supposedly wise man. Oh, there's in italics and underlined, by the way. Staphylococcus aureus is totally descriptive. Staphylococcus means it's cockeye, or like a ball. Staphylo means it looks like a cluster of grapes. So staphylococci looks like grapes, cluster of grapes. Aureus is meaning that it looks golden. All right, any question about that? Now, when you're naming something in the binomial naming system, after the first use of a scientific name, the name may be abbreviated with the first letter of the genus and then the specific epithet name. So the first time you use Staphylococcus aureus, you have to spell it out. But then all future times you can call it S. aureus. And that is italicized if you can't see it. And Escherichia coli, you're supposed to spell it out the first time you use it, and then all other times use E. coli. But E. coli is so ubiquitous that, and everybody knows what it is, that most of the time, most people just use E. coli, okay? It's found in the colon and the large intestine, and Staphylococcus aureus is found on your skin, as well as your nasal passages and maybe in your throat. We'll talk more about Staphylococcus aureus because it can be a human pathogen. All right, so that's the naming system. Now let's move into the classifying of microorganisms. we classify all living things just to group them together with their similar grouping, meaning if an organism is similar to each other, we put it in the same group. Like dogs are similar to foxes and even more similar to wolves. So we put it in the same, I think they're in the same family. I would have to look that up. I don't know. Certainly dogs and wolves are in the same family. And I think foxes are, but it might be a group order. They might be a group. No, they might not be the same genus. I bet they are in the same family. Anyways, we classify things, and when we're talking about classifying of microorganisms, the first division you'll have is simply all living things. And then we classify things into the 3-domain system. So we're going to talk about classifying all living things on the planet Earth. And the first classification system is the 3-domain system. Blow that up a little bit. The 3-domain system is sometimes called the tree of life because it branches sort of like a tree. There are 3 domains. Domain bacteria, shown here, which I imagine you can't see that. Let me get that. There we go. Now you can see the bacteria there. That's the first domain. The second domain, the domain archaea, you could also call that archaebacteria. And then the domain eukarya. An organism falls into one of these 3 domains based on genetic data, meaning we sequence the DNA of an organism and we can classify it into one of the 3 domains based on how similar its sequence of DNA is to other organisms in that domain. So all of the organisms in bacteria have a similar sequence of DNA, and that sequence of DNA will be much more similar for any organism within the bacteria than to any organism out of this domain, either in the domain eukarya or the domain archaea. And this, when it was first discovered, was quite a shock to the scientific world because we knew about the archaea, but we thought they were prokaryotes and we thought they were the same as bacteria. They were just a special grouping of the bacteria. And when we sequenced them, it was quite apparent that they were much further, well, actually, they are essentially 3 equal domains. And in reality, the domain archaea is a little bit closer, just a tiny bit, closer to the domain eukarya than they are to the domain bacteria. Meaning they're essentially 3 equal domains, but the archaea and the eukarya are a little closer together than either of the other 2 domains are to the bacteria. All right, any question about that? So if anyone ever discovers a new living life form, they sequence the DNA of that life form, and then they can determine what domain they go in just based on genetic data. That's really good because that means nobody ever argues about where an organism goes. You just sequence the data and we know where it goes. And there's no argument about, nope, it belongs here, nope, it belongs there. When we're talking about the domain eukarya, we should mention that there are different kingdoms within the eukaryotes, such as the kingdom Plantae, the kingdom Animaliae, the kingdom Fungi, and the kingdom Protista. Okay? I think you all know about plants and animals and maybe even fungi. Those are the eukaryotes, and we divide the eukaryotes up into kingdoms. And there is a kingdom for all of the bacteria. So bacteria is both a domain and a kingdom, meaning The kingdom is underneath the domain, and then we, for the eukarya, separate the domain eukarya into 4 kingdoms: plants, animals, fungi, and protista. The domain bacteria only has 1 kingdom, kingdom bacteria, and the domain archaea only has 1 kingdom, the domain kingdom archaea. And so it's acting as both a domain and a kingdom. So that our system we're using, and I think the textbook uses that too, is the 6 kingdom approach. But there's no definition of what a kingdom is. And so there are arguments as to how many kingdoms there are. And in fact, scientists are currently in disagreement and have been now for over 15 years, meaning they've never come to an agreement as to how many kingdoms there are. Some people use the 4 kingdom approach, or excuse me, is that the 3 kingdom approach? Yeah, some people use the 3 kingdom approach, meaning kingdoms and domains are the same. Some people use a kingdom approach between 4 and 6, and I'm not sure how that's split up. The 6 kingdom approach, we will be using that, meaning 4 kingdoms in the eukaryotes, and then 1 for archaea and 1 for bacteria. And there are other kingdoms much higher. You can get a 9 kingdom approach. I think there's a 12 kingdom approach. There's a 20 kingdom approach. And what you're doing for most of those is this kingdom protista, it's divided up into more than 1 kingdom. And there's a reason for that, and that is when we divide the domain eukarya up into the kingdoms, there's always an agreement as to what is a plant, and what is an animal, and what is a fungi. But the protista is just everything that's a eukarya that is not a plant, not an animal, and not a fungi. So we always knew it was not a monophyletic kingdom. Everyone understand that? And on this division here, there's the fungi, there's the animals, there's the plants. So on this division here, you could divide the protista into 1, excuse me, my mouse died, 1, 2, 3, 4, 5, 6, 7, 8, 9 different groups, 9 different kingdoms. Now we won't use that approach, we'll just use the 6 kingdom approach because, well, it's easy and that's what your textbook is using. Any questions about that? The point is, there's no definition of what a kingdom is, and so there's disagreements as to where an organism falls and what a kingdom is. With the domains, there's no disagreement, it's defined. Any question about that? All right, let me end this. Move on. Let me state that your textbook says that the 3-domain system has 2 cell types. There's a cell type of prokaryotes and there's a cell type of eukaryotes. And you could call that a super kingdom, larger than a domain, meaning you divide all living things into 2 super kingdoms, and then the 2 super kingdoms are divided into the domains. But your textbook isn't calling it a super kingdom, it's calling it a cell type. The bacteria, all of this domain has 1 cell type, and those are prokaryote cell types. The domain archaea also has 1 cell type, and that also is a prokaryote cell type. The domain eukarya has 1 cell type, and it is a eukaryote cell type. Okay, any question about that? So these 2 domains have prokaryotic cells. This domain, which is pretty obvious since it's given the name eukarya, as eukaryotic cell types. All right? And the cell type is defined. We will talk about that later. How you can tell the cell type is you just look to see if there's a nucleus and whether they have cellular organelles. I'm talking about membrane-bound organelles. And if they have a nucleus and a membrane-bound organelles, it's a eukaryote. If they don't have those things, then they are a prokaryotic cell. So the cell type is defined the same as the domains are defined. It's just after that, in the classification system, there's no agreement on how to define kingdoms. And we're not going to talk about classification at any level, at least now, at any level below the kingdom. Okay, you can classify things into lower levels, getting down to the species name, and I showed you the species of Homo sapiens. And you could even go further than the species. You could say the strain or race. All right. Any questions about domain and cell types? If not, let's move on to the characteristics of the different domains. So when we're talking about the domain bacteria, we'll talk a little bit more about this one because we're going to study a lot about the bacteria. The cell type, as I mentioned, were prokaryotes, so they have prokaryotic cells. You can define the bacteria. Okay, somebody, you have a microphone on, somebody who just recently connected, could you turn your microphone off, please? You can define the bacteria by, they have peptidoglycan in their cell wall. You're going to define them, although this is true of the archaea also. They multiply by binary fission. For energy use, the bacteria can get their energy source from essentially any source. They can get their energy source as an organic chemical, and we do that. We get our energy needs from glucose in the food we eat. Just a minute, people, I've got to silence somebody. And I can't do that unless I'm not sharing. Come on. There we go. There we go. Sorry about that. I'm afraid if you were watching, you could see who I muted because of, you could see what I was doing. Actually, you couldn't see, so I wasn't sharing at that point. Okay, but there are bacteria which can also get their energy needs from an inorganic chemical. That could be something like iron or hydrogen sulfide gas. And then there are bacteria which can engage in photosynthesis, so they're getting their energy needs from light. Most of the bacteria are free-living decomposers or primary producers, but there are some that are parasitic, and some that cause disease in humans, so we call them pathogenic. If a bacteria causes human disease, it is a pathogen. Okay, any question about the bacteria? If not, let's move on to the domain archaea. They also have prokaryotic cells. I'm not going to ask question, are any of them having peptidoglycan? For this class, we're going to say that the archaea do not have peptidoglycan in their cell wall. And I should go back to the bacteria and say that if the bacteria have a cell wall, they have peptidoglycan in the cell wall. And that separates out the bacteria from the archaea as well as the eukaryotes. Some of the archaea are found in extreme environments. We call them methanogens, and so they'll be found in an environment where the bacteria are producing methane gas. That would be poisonous to us and many bacteria. You can find the archaea in extreme halophiles, meaning in high salt concentration, like you can find archaea growing in the Dead Sea between Jordan and Palestine. You can find them as extreme thermophiles, meaning they're growing in very hot environments, which that would be toxic to eukaryotic cells. But if we look further, and your book doesn't discuss that very well, you can find the archaea in all of Earth's biomes. They're a significant portion of the soil microorganisms and a significant portion of the microorganisms found in seaweed, in ocean water. Can anyone guess, are any of the archaea pathogenous? Does anyone know? Anyone done the reading?
They're not, right?
Yes. The archaea are not pathogenic. To our knowledge, there are none of the archaebacteria or archaea which cause human disease. And for that reason alone, after this lesson, we will not really be talking about the archaea. And that's because in this class, we're going to focus in on the prokaryotic cells which cause human disease, and none of them are archaea. Okay? All of them are in the domain bacteria. All right, any question about the domain archaea or any questions about anything? All right, if not, we'll move on to the domain eukarya. The domain eukarya, I mentioned, has 4 eukaryotic kingdoms, Plantae and Anneliae, fungi and Protista. All of them lack peptidoglycan in their cell wall. If they have a cell wall, and of course animals do not have a cell wall. Plants and fungi do have a cell wall. Protista, some of them have a cell wall and some of them do not. We'll talk about that kingdom a little bit later. The first kingdom I want to talk about is the kingdom fungi. Fungi is plural. If you want to talk about it's singular, you use the word fungus. Let's see. The only thing you need to know about this kingdom is really that it has the molds, which depending on who you ask can be called multicellular, which when you're looking at mold on bread, it looks multicellular. But you could argue that if you take one cell off of this mold and grow it in a good environment, it can reestablish the mold colony. So one cell is all you need, so you can say it's unicellular. We won't get into that argument. Also in the kingdom fungi are the mushrooms, which everyone agrees are multicellular. And you know what that is. Some of you probably eat mushrooms. And then the unicellular yeast. Here we're looking at yeast. They're important in rising bread and brewing beer and wine. The fungi generally are important decomposers. Most of them are decomposers, meaning they decompose things that used to be alive, and that's how they obtain their nutrients. But there are a few, mostly the yeast, which are pathogens, and they can cause human disease. Okay, so some of the fungi, particularly among the yeast, can cause disease, and those are pathogens. The kingdom Protista, the protus, is the kingdom that we will talk about. And for this kingdom, we'll divide it into 3 groups. Like I said, you could divide it into 9 groups if you wanted to, based on DNA. There's the kingdom, well, not the kingdom, it's the kingdom Protista, and it has a division, and some of the algae are Protista. These are the plant-like Protista. They engage in photosynthesis, and I'm only saying some of the algae. And if you ever go to the Oregon or Washington coast and you see the algae washing up on the coast and you pick it up, that's multicellular algae. What we're talking about is that some of the algae are singular celled or very tiny, and you have to use a microscope to see it. And those are the plant-like protists, and those belong to the kingdom Protista. Okay? What you need to know about them is that they engage in photosynthesis to make their energy, and they do produce molecular oxygen just like a green plant, and organic compounds, largely glucose, but they can build other things from glucose. Anyways, those are the algae, the plant-like protists. We won't talk much about them in this class because they do not cause human disease. Any question about the algae? The next part of the kingdom Protista that we're going to talk about are the protozoa. Some examples: amoeba, paramecium, euglena, and there's an amoeba. They absorb or ingest organic chemicals, usually by putting pseudopods around them. Actually, that's only of the amoeba. The others don't have pseudopods. Sorry, I'm getting a little confused here. The amoeba have pseudopods. They do ingest organic chemicals, and they have various ways of doing that. Some of them actually have an orifice, sort of like a mouth, and ingest it that way. Some of them are motile, and they can be motile by pseudopods, where the cell, if it's moving this way, puts its cytoplasm out in a pseudopod and then pulls it up from this direction, and then the cell slowly moves that way. The amoeba move that way. Some of them, like the paramecium, they move by cilia. Those are little hair that beat and move the cell in the direction that the cell wants to go. Others of them have flagella, and we'll talk more about flagella because that's the way bacteria move. The human sperm cell has a flagella. Some algae have a flagella. Let me think if I can remember another cell that has a flagella. Well, some of the cells in your throat have flagella, but they don't move with the flagella. They move the mucous membrane. Anyways, then you could divide the protozoa up on how they move, and we're not going to discuss that, at least in this lesson. When I talk about the parasites, if we get to it at the very end, the last lesson, we'll talk more about that. The protozoa, some of them are parasites, and if we cover the last lesson, we will be talking about the parasites. Any question about the protozoa? Okay, the last group of the kingdom Protista are the fungus-like protists, and obviously they act sort of like fungi, and none of them cause human disease, so we're not gonna talk about them. Boy, that's really small. I just wanted to make sure I didn't have something at the bottom of that I couldn't see. All right, any question about the kingdom Protista? If not, let's talk about the kingdom plants. The kingdom plants, you can divide them into the mosses, the ferns, the conifers, and the flowering plants. All you need to know about that is all plants are multicellular. We're not going to be studying the plants, and that's because they do not cause human disease. Any question about that? All right, let's talk about the kingdom Animalia. You can divide the animals into the sponges, the worms, and scientists have another name for worms. They call them helminths, the insects, and the vertebrates. All animals are multicellular. And guess what? Some of the animals cause human disease, and we will study them. Anybody know what animal causes human disease?
Insects and worms.
Could you say that a little louder?
Insects and worms.
Actually, we're going to study the worms, the helminths, because that's what microbiologists study. Insects can bring disease to humans, but unless you have like a flea infestation, which that's not really a disease, insects do not cause human disease. They can bring disease to humans, like malaria spreads to humans because of mosquito bite. But mosquitoes do not cause disease, don't start the disease on their own. They have to bring the malaria to humans. So insects can bring disease to humans. So you got that right.
What about birds?
They don't cause human disease either. They can be a carrier of a human pathogen, and then the pathogen can move from the birds to the human. Okay?
What about rodents?
The same thing. They don't cause human disease. People have pets that are rodents, but they can have a disease-causing organism, and that disease can move from the rodents to the human.
Can't just dogs and cats cause disease in animals? Or in humans?
Dogs and cats don't cause disease in people. They can have a microorganism on the dog or the cat, or in the dog or the cat, which the person can get. So the microorganism can cause the disease in both pets and in people. But it's the microorganism that causes the disease. Okay? And that's true for all of them except for the worms. And there are worms that cause disease when you have a tapeworm, for example, or a worm infestation. That is considered a disease, and the microbiologists study that, and we'll talk a little bit more about worms. And worms obviously are not microscopic, or at least I should say most worms are not microscopic. There actually are a few that are, but their infective stage is microscopic, and microbiologists study them for two reasons: because the infective stage is microscopic, as well as they actually cause human disease. Okay, any questions about any of that? If not, I'm gonna take a 15-minute break, and we'll come back at 2:30. All right? Oh, remember to come in on your second lecture.